Great. So yeah, we're going to continue from where we left off yesterday on LD Score regression and kind of finish off this piece on partitioning and then get into what's listed on the agenda there with talking about genetic correlation. So where we had left off yesterday, if you may recall, is we started with our derivation from a set of assumptions about a basic additive model that got us to kind of the original univariate version of LD Score. And then at the end of yesterday, we had generalized that to now, instead of using a single LD score that related to a single estimate of SNP heritability, or heritability per SNP at least, for whatever we've included in the model. We generalized that to now we have different LD scores for different dimensions of annotations that each have this tau parameter that is essentially a contribution to total heritability per SNP that is affiliated with a particular annotation. And so much in the way that the kind of our initial statement about LD score regression was that once we have these LD scores and their relationship to our expected chi-square statistics from a GWAS, then we can do a regression to get estimates of that heritability per SNP type slope parameter and this intercept here that relates to confounding. Similarly, we can do multiple regression in the partitioned heritability case with our set of different LD scores that relate to different annotations and get estimates of these taus for kind of differentially how much heritability per SNP signal are we seeing in the GWAS in different annotated categories. And so picking up from there, kind of the first thing to discuss for today is what annotations might we be interested in paying attention to as having different taus. And so a ever-growing list of things have been used in this framework, but some of the initial things that proved successful and were a focus of kind of initial motivations for partitioned LD score were things like functional genomic categories. So we started in the toy example talking about coding regions and DHS, these DNA hypersensitivity sites, kind of places that we might, from our knowledge of just genomic structure, might expect there to be more meaning to genetic variation happening there versus in places where we're less sure what that DNA does. And similarly, we can think about promoters and enhancers and other regulatory elements. We can think about sections of the genome that seem highly conserved across different species. So we can regions of the genome where the sequence is very, very similar between humans and primates and other mammals, and go further back, like at reptiles, and sections of the genome that maybe are more important by evidence of the fact that they have continued to stick around. So maybe they're doing something kind of fundamentally important. And so we can say all those things about sections of the genome in that sense, and then we can also start thinking about regions of the genome where we have statements from like other wet lab biology as far as what their activity seems to do. So regions of the genome that we know from, say, EQTLs are affecting gene expression, perhaps in particular tissues or cell types, or that have various histone marks or methylation patterns in the genome that are related to various things. And so these are all kind of spaces of things that people have thought about in terms of where we might want to both allow the flexibility in the LD score model to account for that we might have pre-existing expectations that would say the genetic signal might be different in these places, and also might help feed interesting scientific hypotheses as far as saying, so we've got a set of loci, can we say that the genetics seems to be more active in, say, genes that are expressed in the brain, or other kind of things like that. So the kind of list of even more narrowly concrete examples that's on the slide here partially just highlights that a lot of this is a process of taking published annotations from the literature that continue to evolve as people work on like the biological side of the genomics and applying those to seeing if they are informative about our GWAS signal. And the last note here is potentially useful in that there is an important step here between like some genomic annotation and how we're going to include it in our model. And in particular, where exactly we want to draw boundaries on regions. So we might be interested in, say, again, like a collection of brain-expressed genes. But when we say that on a like heritability per SNP basis in this like set of joint conditional effect sizes, do we really mean like just the SNPs in the exon? Do we mean the SNPs that are anywhere within the transcript, so from the start codon to some canonical end codon, though there's also questions there about which transcript you would use. Do we want to include things that are close, like right at the boundary, so imagining that SNPs might affect splicing or start of transcription or things like that. And so often we will, with these annotations, make some statement that we're taking the kind of some canonical version of the genomic annotation and including as part of our modeling some additional buffer region around it, so that things like, say, within 5 or 10 kb of the defined annotation are included as part of that annotation for the purposes of generating our LD scores. So those are possible annotations. How do we want to evaluate the results from LD score regression for the annotations? So there's kind of two main ways that have emerged to go about this. The first is perhaps the obvious one from how we've structured the regression, which is just that we have these estimates of taus for each annotation that are this contribution of that annotation to expectations of total heritability per SNP, that variance in those conditional effect sizes. And so we can just look at whether that tau is significant for a given annotation. But because annotations overlap, we may also be interested in making statements about enrichment for a given annotation, irrespective of what else is in the model. So being able to say that, like, we're going to include all these diverse features about histone marks and coding regions and hypersensitivity sites and, you know, many, many different features. But when we look at, like, is there enrichment in the coding region, we really want to say, like, regardless of whether it's better explained by some, like, transcriber marks or whatever, just tell me, like, overall, if I took the coding region versus the rest of the genome, how enriched for heritability is that coding section, regardless of what the model says about the taus that get you there. And so the common metric looking there is to look at this idea of the SNP heritability for a given annotation. So this generally only applies in the case of binary annotations, but heritability per SNP within that annotation. So for category C is the notation that gets used in the papers, relative to the total genome-wide SNP heritability per SNP. So just kind of a fold change in what that per SNP heritability is looking like. And so I promised you we'd kick off today with real results, so let's look at real results. So starting with the functional annotations, so this is in the first paper from Hillary with this method, looking at really things around coding and conservation and some enhancer marks and such. And so these are results where they took for some of the well-powered GWASs that existed at the time, and saw by and large that significant, this is in terms of enrichments, so significant enrichments in coding regions, even stronger enrichments in conserved regions. I think there's previous discussion that also deflation in these annotated as repressed regions based on lack of transcriptional activity. But these aren't saying that there's zero heritability here, just that it's less than average genome-wide heritability per SNP. And then kind of a more hypothesis-driven thing that, in particular, there's this class of enhancers called phantom 5 enhancers that are expected to be related to kind of immune traits. And indeed, they showed strong enrichment in a meta-analysis of different immune phenotypes.
Raymond?
Yeah.
I'm sorry if I sound like a broken record, but just going back to Robot's question yesterday. So the reason, so we see these massive enrichments for two of the categories here.
Yep.
But the reason that it doesn't make that much of a difference if you incorporate that into the priors when you make the score is that the SNPs are just a tiny share of the genome. Is that?
Yeah. So I've got another slide that'll deal a little more directly with that point. But yeah, so you can imagine that, like, taking coding as an example, that we're talking something like a 5-fold enrichment in heritability per SNP, but that's a 5-fold enrichment that only applies to, as this annotation was defined, about 1.5% of the genome. So if we recall, exome is roughly 1% of the genome. This is once we've added a buffer region and are allowing, like, introns to contribute and such. So, you know, 5-fold extra from that 1% is still a small slice of the overall heritability.
I just have a question about the overlapping of annotations.
Right. Yeah. So yeah, it's that. For example, of conserved regions, many of those are places that are also coding.
That's just like that it doesn't have it
I suspect there might not be overlap with the other categories that are on this particular plot, but certainly they could overlap with other statements about like histone marks and such. And we're going to talk in, I think that literally, no, 2 slides from now. Will pretty much always overlap if we've defined a continuous annotation that's talking about allele frequency or something. So this kind of accounts for, so if that model wasn't used in these results, but we can imagine if we fit this model where we also had, say, some separation of SNPs by allele frequency to try and loosen up our assumptions about what that looks like, that this enrichment could reflect that if SNPs in coding or conserved regions tended to be rarer than SNPs in other places, and if the effect sizes of rare SNPs tended to be larger than of the common SNPs, kind of beyond like a equal per SNP heritability expectation, then even if the like driving feature wasn't that the effect sizes are larger because they're conserved, that was like better explained in our model by just allele frequency, it would show an enrichment here because we're looking at just the overall enrichment that kind of collapses across any degree of overlap for those SNPs. Yeah.
Please tell me if I'm completely wrong, but would it be possible that enrichment of like all the nearby regions would lead to potentially upward or like overestimation of the estimates?
I'm not sure I quite follow the question. So you mean that like is the question like would it potentially that you'd see an enrichment in some annotation that's not due to that annotation, but because it's like nearby something else that does actually matter? Yeah, so that's certainly a possibility. And so to some degree, when we choose to look at enrichment, we're choosing to accept that possibility that it may have nothing to do with, say, conservation. That's like why those SNPs have larger effects. It just happens to be a good indicator for some set of SNPs that happen to be in the right places. That's the reason that we may also be interested in looking at those taus, where at least if we've got the other things that we think are relevant that relate to overlap between these different categories, it might help us distinguish whether one particular annotation is actually explaining where that higher heritability per SNP is coming from as opposed to other nearby annotations. Cool, so that's functional annotations. Similarly, we can look at, I mentioned talking about looking at annotations related to particular tissue types. So this, the paper from 2018 did this on gene sets defined based on expression of the genes in various tissues. This has also similarly been done based on chromatin information for transcription in various tissues. And so the set of plots that was used here is that each of these, each point in the plot here is p-value for the test now of tau for the annotation that is related to expression in a given tissue, conditional on a model with all the functional conservation type things. So this is like against a background of general like non-tissue specific statements about functional genomics. Is there significance on top of that within this joint fit of the taus for genes that are related to expression in a given tissue? And so the points here are colored by kind of general categories of tissue, and then you get more specific tissues or cell types as the individual points here. So for example, in schizophrenia, you've got a large number of tissues or cell types from the brain and other parts of the central nervous system that are all have significant taus in this model based on data coming out of, for example, psych code. And kind of as implied by that collection of points, you can push this further in terms of if you want to define your cell-based or your expression annotation is more narrowly like, we got a signal in brain. Do we see specifically enrichment in cortex, or specifically in neurons, or specifically in certain classes of neurons. So lots of room to decide how you want to define that annotation that might match a scientific question. So that's categorical annotations, and I mentioned before the potential for continuous annotations, which are a little harder to come up with nice visualizations for, but at least the approach that's been taken thus far is to kind of, even though the fit for the model in the annotations that the annotation value is continuous, for display purposes, you can think about dividing that continuous value into quintiles for kind of the distribution of that annotation across SNPs, and then think about the proportion of heritability that is attributable to SNPs that are in a given quintile. So the results here are from applying this to a number of traits and meta-analyzing those results down to summarize. But so things that have been looked at in kind of the first approach to this was things thinking about like nucleotide diversity. So we've talked a bit about like regions of the genome that are highly repetitive, and it's not clear like what it matters if you've got a string of GC for like the 1,000 base pairs of just repetition. And so thinking about whether the places where there's less variability maybe matter more or less in terms of mutability considerations, similarly related to selection considerations. We've talked some about assumptions about allele frequency already, so being able to break that down more continuously in a way that we're not just assuming a relationship between effect size and allele frequency as well. So that's kind of what that looks like. And so this was a meta-analysis across a small group of traits. We can also start thinking about what this looks like. Again, going back to those results in UK Biobank. So this is having run the partitioned model with all those functional categories and some continuous annotations on each of the significantly heritable traits from our analysis of UK Biobank. And so you can see that there's lots of signal in these taus for many of the annotations. Yeah, go ahead.
So I understand. So is it not a phenotype? Is that
Yep.
And so you're just looking at the so like we look at the red conserved dots.
Yeah, so like this dot is for a single phenotype, the test of the tau for the conserved annotation. This is for a different phenotype, the test of the tau in partitioning for that and so forth. So the just for this figure is just to say that kind of on average across traits, we see the strongest conservation related to things like CG content, and GERP is a kind of predictive annotation of likelihood of functionality that pulls from different conservation metrics, among other things. And that actually the same conservation thing used previously, and there's other histone marks and such that show up highly here as well. This is from an earlier version of the analysis than we have now. You can find an interactive figure at that link that will let you hover over the individual points here and know what phenotype is going with which dot and all those kind of fun things. But yeah, just to get a sense of that, at least in a sufficiently large sample size, it is quite normal to see significant taus for a lot of these annotations. And that's perhaps meaningful in the sense that one of the ways we motivated the partitioned results is that this is generalizing to get away from some of the simplifying assumption we made in the univariate model. So one thing that's perhaps noteworthy is, so if we take from having done this partitioned LD score approach on the phenotypes and compare it to if we had done just the univariate version without accounting for these annotations, and see what differences we would see in terms of our estimate of SNP heritability and in terms of our intercept, what we find is that things are highly consistent. So the kind of univariate estimate is not doing too poorly despite the fact that it didn't have all this additional information. Adding that additional information is like it's significant in terms of knowing about those annotations, but it's not making a huge difference to our inference about overall SNP heritability. But especially for things that have the clearest signal of total heritability, you do see a little bit higher estimates if we have been able to account for this additional variability. So just a general better model fit as far as how much of those chi-square statistics can be explained by our LD scores once we allow for this additional kind of variation across annotations. And so consequently, related to that, our estimate of the intercept in terms of how much confounding is left in the GWAS is lower once in the partitioned model than it is in the univariate model on average, because at least some of that intercept that we see in the univariate case is not because there's confounding, but it's because our simplified set of LD scores aren't fully capturing what that variation in the chi-square is coming from. And so in the classic regression sense, the kind of essentially error in those LD scores is turning into an increase in our intercept term. And so we would potentially overstate how much confounding we were facing in our GWAS if we didn't try and take advantage of more information about the partitioning. A couple final things to wrap up here on the partitioning. That mostly stuff we've already covered, so just that code and LD scores, especially the LD scores related to many of these annotations that have already been published on, are available. Sample size demands are high. We'll talk about that a little bit more in the genetic correlation section. So essentially, if you don't have enough signal to be confident about the overall amount of SNP heritability, then partitioning that is kind of a lost cause. And partitioning that also faces a challenge of, at some point, you're, especially due to us relying on block jackknife standard errors for LD score, if the annotation is a sufficiently small slice of the genome and in sufficiently few locations, accurately thinking about the variation of that annotation across the genome is highly challenging in this framework. So similar to the previous statements about how the kind of SNP heritability behaves best in more polygenic contexts, we also want annotations that have broad dispersal across the genome to have a decent chance of fitting well in this approach. And then the last piece I'll flag, especially in thinking about ideas about these taus and these enrichment values, is that any time you're talking about an enrichment of signal in some given category, that's an enrichment relative to some statement about what the baseline expectation is. And so in particular, this is shown up in the literature that there's another set of methods that branches a bit more directly off of GCTA, though the sum-her method is at least a little closer to LD score, that makes different assumptions about what the distribution of effect sizes might look like with relation to LD block structure. And so if you fit that model with nominally the same set of annotations, you'll see very different enrichment results because you've set a different baseline expectation about what like the normal behavior of the effect sizes is. And so interpretation of the enrichments is very much attached to the choice of model that we've made here as far as what expectation is for heritability per SNP. And then kind of on that same point about enrichment, that actually goes back to what David flagged, is that the enrichment relative to baseline is also not necessarily a statement about contribution to total quantity of heritability. So just because a annotation is enriched doesn't mean it's the most important thing in terms of where the majority of the SNP heritability is coming from. So this is an example that was used in the Pritchard Lab omnigenic model paper, but is a useful observation that for example, in schizophrenia, if we take the enrichment here in different tissue types, we see that the strongest enrichment is in annotation of expression for, say, frontal cortex in the brain. But if we were to think about it in terms of proportion of total heritability explained by SNPs that fall into different categories, the plurality of the heritability is actually more associated with regions of the genome for genes that are just broadly expressed in all tissue types, because there's more of them and they are at least somewhat enriched. So in terms of thinking about fractions of overall heritability, it's a larger slice, even if they're not as enriched on a heritability per SNP basis. So again, that kind of idea that base rates and baseline models matter a lot to thinking about these results. Any other questions on partitioned model before we move on to genetic correlation? Yeah.
Maybe not the most statistical question, but one of the things, it seems like this is just kind of like serving as a check on kind of our own intuitions, being like, oh, this thing that is associated, or this phenotype that is associated with height seems to be expressed in things like bones. I'm wondering if there are any good examples that you or other people know of like some more specific utility that's been gleaned from these results, rather than just saying, oh, it's a brain thing and we found brain expression or brain engagement.
Yeah, so the question is to what extent are there results that aren't just confirming our own intuitions for the sake of the Zoom there. So I'd say, so I guess having the results that confirm our intuitions is useful as a baseline, partially just because like, face validity of the results is useful as a starting point, especially when you're talking methods that are still getting their feet under them and understanding whether the annotations are actually picking up signal we might expect. But in terms of places where it's produced results that weren't necessarily certain at the outset, I would say a couple different examples. One is in talking about some of those like tissue cell type kind of results, the kind of further down in the granularity those get pushed about being able to look at gene sets that are specific to an individual cell type or to a class of cell types. The kind of weaker the pre-existing assumptions or knowledge about those results were. So there's hints of, though, kind of at the boundaries of power in at least the kind of published version of the analysis, about differences between, say, GABAergic and glutaminergic neurons and their functionality in schizophrenia and bipolar in education. That while we probably could have, would have trusted just a priori statement that brain or even neurons were important in those phenotypes, we probably weren't as certain at the outset about kind of hypotheses about specific subtypes of neurons. So there's that kind of granularity push where you pick up new information. And I think this is less my field, so I'm having to go off of other people's statements, but I think some of those continuous annotations around things like selection metrics and MOF and LD in different populations, nucleotide diversity and such, that there were, at least for some of those, there were competing hypotheses where, depending on who you asked, they would have given you a priori answer that differed from somebody else's assumption about what was true. So there has been room there to test people's assumptions. So at least those are the two things that come to mind off the top of my head. Cool. All right. So moving forward ahead now into genetic correlation, which is the last of the three set of papers here that I said we were primarily working through methods from. Though if you prefer review articles rather than original methods papers, there was a very nice review article talking about not only LD score regression genetic correlation, but also just the concept of genetic correlation in general and utility of thinking about it. But so motivation as we move to this idea of genetic correlation is that we talked all this about kind of signal and GWAS of individual traits, but we have GWAS of many different traits. And kind of the naive observation is that we know that phenotypically these traits are correlated. And we know if we start eyeballing the results from these GWASs or thinking about meta-analysis across sets of related traits, that we can certainly start finding loci that seem related to multiple phenotypes. So this particular result is out of cross-disorder analysis the PGC did of different psychiatric phenotypes. So finding loci where, relative to the power that existed at the time, a loci that we didn't have genome-wide significance yet for association of the SNP with any of these individual phenotypes for ADHD or schizophrenia or depression and such. But did, if we've been analyzed across those traits, see signs that there was probably there at least seemed to be some shared effect of the SNP that related across these phenotypes. So maybe signs of pleiotropy or the hypotheses like that. So kind of relative to this kind of qualitative observation, or at least the quantitative evidence for individual loci that seem shared. We might want to make statements about kind of similar to way we'd like to summarize the amount of total heritability attributable SNPs. We might want to say something about the overlap of the genetic factors between pairs of traits. And this is nice both in the sense of looking at how much the genetic evidence links between the two. So getting beyond just the idea that the phenotypes are correlated, but is the part of that phenotype that's associated with the genetics correlated. And also a point that came up earlier in the week, I think during Paige's talk, that in some cases being able to do this comparison with the genetic information will get us around some degree of epidemiological confounds, or just the inability to look at a pair of phenotypes easily at the same time due to the difficulty of getting data collection that captures both of the phenotypes in the same individuals at the same time. And so with that motivation from thinking through how we get this back into a statement from LD Score, and you can start back at, again, our same additive model, and now we're just thinking about what that model looks like for two different phenotypes, where each of these have their respective effect sizes. So we'll say, yeah, I know. I just realized I think the same issue came last year, and this is the one notation thing I haven't fixed. So yes, this is intended to be a different phenotype and is not meant to be related to the set of covariates here. So yes, pick your favorite non-Z letter and plug it in here in your mind. Right, yeah, sure. But yeah, so we have our two different phenotypes. They're related to the same universe of SNPs via some set of these conditional effect sizes. And we have our same set of covariate confounders and respective error terms. And so with this model, the kind of first thing we might want to figure out is how are we going to define a genetic correlation parameter? So we defined heritability previously in terms of the kind of proportion of the phenotypic variance attributable to the genetic piece of this model. And so how are we going to define genetic correlation? So one way we could think about it is, well, we've got effect sizes for the SNPs here and effect sizes for the SNPs here. So let's just talk about the correlation between those effect sizes. Fairly straightforward, fairly intuitive, just like, are the effects correlated? But we could equally develop an intuition where we're interested in the correlation of the overall genetic component between these two. So how much is this total contribution from the genetics, the X beta, and the total contribution of the genetics for the other phenotype, this X alpha, how much are those overall quantities correlated? And so you can work through, as was nicely done in the AA paper, and so credit to this clarity of thought on the differentiation between these goes fully to the SSGAC. The difference here in terms of the quantities that you end up estimating is that essentially the correlation structure of the SNPs, so LD matrix omega here, now influences our thinking, and in particular, the idea here is that we could imagine a cartoon example where for our first trait, all of the even-numbered SNPs in the genome, so SNPs that like even-numbered base pairs have effects on the first phenotype, and for our second phenotype, all the odd-number SNPs in the genome have effects on the phenotype. And by LD, the like neighboring SNPs, haplotype structure, we've got near perfect correlation in terms of, say, prediction about those two phenotypes. But the actual joint effect sizes are completely disjoint, and there's no correlation there. So it's possible that depending on what we're interested in for the purposes of thinking about what it means for a pair of traits to have correlation of their genetics, that we might be more interested in this quantity. And we could actually carry that thinking a step further and say that instead of thinking about the correlation of the overall genetic component or correlation of those per SNP conditional effect sizes, we could just be interested in the question of how correlated are the two GWAS results going to look. Reflecting on the idea that showed up in our initial derivation of LD score, that the effect size we see at the GWAS level, those kind of marginal effects associated with a given SNP, are related to those conditional effect sizes by way of kind of convolving them with the LD structure, which would get us a further progression where now that LD structure is even more important to our thinking about this quantity, and would be able to tell us something about how similar the GWASs look that is distinct from these either two ways we could think about correlation. Yeah.
I'm sorry, this may just be complete lack of knowledge here, but how did we get from, so we're just taking just the beta GWAS is just the marginal, so it's just the derivative of that term? Right-hand side term, same term?
Yeah, so you're asking about how we get to this relationship here?
That one, like, I get, like, we just want to estimate that quantity, but to estimate that like, how does the answer of that, like, is the thing on the right-hand side?
I see. So where is the far right side coming from here?
Yeah.
So
Sorry,
yeah, no, it's totally fine. So this is essentially just a definitional statement once we've, of terms of writing out an expression for the correlation, assuming mean 0 on the effect sizes. So this is so it's perhaps easier to walk through up here. That you just having the kind of cross product across SNPs relative to the variances within those SNPs as far as a standard definition of correlation matrix form. The reason for representing it this way rather than just talking through what we mean in terms of correlation is to see this relationship that essentially the kind of as a computational statement of the difference between these values, the key that emerges is that by inclusion of the genotype in this definition, you end up with attachment to LD, and by the GWAS effect sizes relating to the kind of what those conditional effect sizes look like after LD, you get kind of a progression forward in terms of how much the LD is mattering to that computation. So this is just a kind of definitional statement of the correlation under an assumption that the effects are mean 0. Okay. Fair enough. You could also just follow up in more detail afterwards. But yeah, the important part here is mostly just the statement about types of correlation we could think of as a genetic correlation rather than worrying about the exact computation here.
I was just with you until the second step.
Okay. That's fair. Cool. So there are many ways we could define genetic correlation, as I alluded to. There are merits to each of these, and they largely turn into a choice of like scientific question of which quantity we're interested in. But the potentially simplifying thing is that under some decently strong but similar to LD score assumptions, these three quantities actually end up being equivalent in expectation. And in practice, the EA paper made the effort to compute at least the first two of these, so that correlation of the effect sizes versus the correlation of of that overall genetic risk, kind of regardless of how that divides amongst the SNPs, and found that in practice, at least for educational attainment, that those values were quite similar, and so maybe we don't need to worry too much. And so in practice, at least as implemented by LD score, the argument for deriving the LD score version of genetic correlation proceeds off of this correlation of total genetic risk version, but ends up with an estimator that is actually estimating based on a definition of the correlation of the effect sizes. So it makes an assumption that makes those two things equivalent. And so with that kind of forecast of what that derivation is going to assume, let's look at kind of the sketch of that derivation. So again, our two phenotypes, I've omitted the covariate terms just for notational simplicity, not because we actually need to omit them. But so our statement about the model is we have our two phenotypes, and now those effect sizes, those joint effect sizes, beta and alpha, have a covariance between them where variance remains proportional to the SNP heritability, and now there's a covariance between them that is going to be our thing of interest for thinking about genetic correlation. So what's written here is the covariance term, but we can easily standardize that by those SNP heritabilities to get to a correlation term. That we have a matching statement about what the kind of residual terms look like, where as long as we're omitting covariance, that variance just turns into 1 minus h squared because of normalization. But we similarly allow for the possibility that there is covariance in those residual terms as well. And we'll get to more details on what impact that has. But so we're going to make a set of assumptions that essentially extends the same assumptions we've made in the single trait case, where we've got expectations of 0 and no correlation other than the correlations that we've stated up here. So again, our genetic effect sizes, the conditional effect sizes are independent of the error terms. The error terms are in those residual terms are independent other than the way that they've been specified up here. And so starting from a definition of genetic correlation as being correlation of these total components of polygenic risk, living out some notation, we can kind of break that idea up that total polygenic risk is the sum over the effect sizes on the SNPs. And so with the mean 0 expectations, that correlation just breaks down into a set of cross products for the correlations of each of those terms. And we're going to make the strong assumption here that essentially all the cross terms are 0 in expectation. So the first part here is the same kind of local independence of effect sizes assumption we made in the univariate case, that knowing the conditional effect size for one SNP doesn't tell you about conditional effect size for any other SNP. So essentially random IID draws as far as effect sizes. And we make that assumption both within each of our two traits, so amongst the betas and amongst the alphas, and also that for different SNPs, you don't get any information across trait as well. So essentially the only place where there's correlation in this random draw of effect sizes is at the given SNP's conditional effect size, that there's a correlation between trait 1 and trait 2.
Where do the beta beta and alpha alpha terms
Mm-hmm.
come from?
So it's that, where do those terms come from? That's a good
I think it
question.
should be there.
I'd have to go back and refine from where I copied this derivation from. I agree that it's not immediately obvious why they're there. There should be. Those are sums, those are sums. I'm going to have to punt for the moment because I'm not recalling off the top of my head how they end up in there. But I will definitely circle back on that maybe after the break. In any case, we get rid of a bunch of cross terms, which is where we kind of escape where we escape out of having genotype in the sums here, that now because there's not cross-term relationships, and that there's not dependent, again, the LD score assumption that there's not dependence between the effect sizes and variation in the SNPs, that these are separable terms. And so functionally, we end up only needing to look at this correlation between the effect sizes that get to our correlation term. All right. And so plugging this back into what we've said previously about the GWAS betas and kind of their statement of what those effect sizes look like relative to these conditional effect sizes. We, instead of thinking about the chi-square statistics as we did before, we now think about the z statistics. Got these as zetas to try and avoid the other confusion with z. But these are, yeah, just standard z statistics in terms of betas and standard errors in our previous statement about what those look like in terms of these effect sizes. And so it turns out that what we get nicely is that expectation for the product of the two z-scores at a given SNP, so z-score for the first trait at a SNP and z-score for the second trait at a SNP, is this product of the previous values. And given our previous assumptions about cross terms, we end up being able to pull apart a term here that is related to sample sizes and the stratification terms, essentially cross product between stratification terms and cross product between error terms that we have not said are independent between the different traits. So we get kind of that stratification-y piece again, and then the piece that's related to the genetic signal, where we have our expectation of the cross product of those effect sizes and the LD piece. And so we're back to a regression-looking equation where we have now our desired genetic covariance that relates to LD scores and an intercept term that does not depend on those LD scores. And so again, we can observe our set of z-scores from the GWAS, observe sample size and compute our LD scores, and get to an estimate of the slope that is dependent upon that genetic covariance that we can back out into a correlation value based on number of SNPs and the heritabilities. Based on defining that, by definition, our genetic correlation will be just the genetic covariance standardized out for the genetic variance in the two traits. All right. So what is that intercept term? What's maybe some intuition for what's happening there? Similar to how we had the intercept term in the univariate case, where there was a piece of variation in those chi-square statistics that are just due to estimation error, and then a piece that emerges from us allowing for there to be some additional variability that is stratification related. Once we're looking at that across a pair of phenotypes, those terms fall through in terms of that there might be some correlation between the estimation error in the two. So for example, in the case of sample overlap, and there might be a covariance of the confounding that exists in the two. So for example, the same kind of population stratification structure is mattering in both of our phenotypes. So we can imagine if we have a pair of phenotypes where, say, the decline of ancestry north to south within Europe is in some way correlated with the phenotype in a confounded way, and that applies to both of our traits, then that same pattern of inflation for different SNPs in the genome, proportional to how they relate to that axis of ancestry, would show up in both GWASs. And so in terms of similarity of the z-scores out of those GWASs, that we would see a term that tracks with that stratification. Cool. But for thinking about the sample overlap piece of that, we could further work through what that expectation for the correlation of just those residual error pieces looks like in the absence of stratification, and express that directly in terms of the number of overlapping samples and the phenotypic correlation between the two phenotypes. That maybe is a little more intuitive to read than this version is in the case where there's no stratification. That really the intercept is keeping track of that potential covariance in the residual terms that aren't the genetics. So all the same caveats about like how this proceeds in terms of fitting and about what the assumptions we've made to get here from basic LD score apply here. We're also, now that we're talking about two different phenotypes or two different GWASs, have an additional piece that we're in thinking about our LD scores. We now need LD scores that we've assumed that LD is the same in both of our GWASs. There are other methods that extend beyond this for if we, for example, are interested in genetic correlation across populations, but at least as derived here, we're assuming that a single LD panel matches both of our GWASs. And then in terms of getting towards interpretation aspects, there is at least a little bit of additional flexibility here where in thinking about the correlation between a pair of phenotypes, that because that statement is going to be standardized out by our statement about total SNP heritability, if we've done something in the course of how we've arrived at the LD score derivation that biased those heritabilities a bit or ends up being a little weird in terms of how we've chosen a universe of SNPs, when we make this correlational statement about the correlation between the genetic effects, the things that just equally apply to heritability kind of standardize out of what we end up with in terms of looking at the correlation. And so we're at least a little more robust to violation of those assumptions. So James Lee and colleagues have a nice paper that show this more mathematically in terms of for particular cases of confounding and kind of stability of the genetic correlation. Same intuition has been applied for defending things in partitioned SNP heritability, though I don't think anybody's managed to show it as formally in that case, but at least some room to be a little more confident in results we see for genetic correlation versus what we see in thinking about meaning of an actual point estimate of SNP heritability out of LD score. But with that defense of the estimation piece, there's still some interpretational questions. So in particular, as with any correlational work, correlation is just correlation, which can be maddeningly vague. We'd really like it to be a pleiotropy statement. Or in some instances, people really want to say, well, the genetics are correlated. Obviously, the only way this happens is causation. But correlation is not causation, and that still holds even if you add genetic to it. The other thing is kind of even beyond just being correlation, like we've only made a statement about like that one bivariate moment. So intuitively, we'll think about correlation as meaning, well, so we had our effect sizes. We've got our like true betas for two different traits here, and if they're highly correlated, then we must see a high correlation. And if they're uncorrelated, then we just got a random distribution here. But given that we've only talked about the moments, there's nothing that says that it's some nice distribution that matches that moment. And so we could equally have had that the correlation actually reflects that there is some like even more perfectly correlated piece that is shared, and then some piece that is unique to a phenotype. And the convolution of those two things averages out to a given correlation. But that's not necessarily the same statement as far as like what that kind of bivariate distribution looks like. And this is perhaps even clearer in terms of when we say that there's no genetic correlation between a pair of traits, that could equally mean that they share exactly the same causal SNPs and just their effect sizes are uncorrelated, which would be this top picture here. They're like, it's the same set of dots, but there's just no correlation of those effect sizes. Or it could be that they're completely disjoint, that the effect size, the SNPs that matter in one trait, only matter in that trait and are completely disjoint from the SNPs that matter in the other trait. And the statement of no genetic correlation says nothing about differentiating between these scenarios. The other practical challenge is that just where, as with all things, and especially bad in the LD score context, we are subject to the power of the two GWASs for our ability to get to any useful statement about the two traits here, and we're subject to that block jackknife structure that if, again,